Good afternoon. Oh, good afternoon, everyone. Welcome to today's side event. Yes, okay. Maybe they're adjusting that downward. So we're going to use the microphones today because they are recording this side event. Anyway, as I was saying, why I have had an opportunity to participate in some of the image meetings and really see the scope and the dynamic nature of this project. And my hat's off to the group that put this together because it's a very, very interesting project. A couple of housekeeping issues for us before we get started today. Why, as I just mentioned, why this session is going to be recorded. There will also be some photographers moving around the room and taking pictures, and they're going to want to make sure that you sign off on the consent forms, I think they have as well, so that you'll know that that's happening. There's going to be a survey handed out, and there'll be an opportunity on the survey if you're interested in receiving the image newsletter, why you can check that box, and they'll be glad to update you on the progress in the project. We've got two presentations today. The first presentation is by Dr. Michèle Tixier-Boichard, who's also the Directress of Animal Genetics and Integrative Biology within the INRA system, and she's also the project coordinator for IMAGE. Her talk is going to give you an overview of the aims and current achievements of the program. Michèle?
Thank you very much, Harvey. Good afternoon, everyone. I'm happy to see various geographic origins, because we will talk about diversity, and we want also to have diverse opinions and diverse answers to our questions. So for this talk, I will just introduce to you the project and the current progress at present, because we are at the mid-term of the project. The project is motivated by considering that animal gene banks represent an asset. As you know, everybody here knows that only a few breeds are not at risk, and we are lacking information about most breeds. But still, governments are investing in gene banks, because a European survey 5 years ago showed that there were animal gene banks in Europe in 25 countries. So it is really an investment of the states, also of the stakeholders. The main initial driver is the preservation, but it could also be used maybe for other support for livestock. And so the initial question we have in our project is, do we get the maximum from our gene banks? Because since they represent a cost and an investment, do we really use them and benefit from them? So to answer that question, we may realize that we are missing knowledge, so we have knowledge gaps. We also have some constraints in the operations of gene banks, and so we would like to bring answers to these questions. And the objectives of the project is to enhance the use of the genetic collections, to upgrade the management of the animal gene banks, and to demonstrate the benefits brought by these gene banks. And there, for instance, some benefits that minimizing genetic accidents, genetic defects, and also allow the livestock sector to respond to new environmental conditions and market needs, because these are resources. So resources, it is both a heritage, but it is also a resource for the future. And how do we plan to do this, to enhance the use and to upgrade the management, is by using genomics, bioinformatics, and reproductive biotechnologies. And so the program ran for 4 years. It started in 2016 until February 2020. We are 28 partners. You will see the map at the end. And for 7 million euros altogether from European Commission. So the main actions that are undertaken in IMAGE are organized in 7 work packages. So the first work package is involving the stakeholders, involving you, but also breeders, also companies, from the beginning of the project to associate the stakeholders all along the life of the research project. Work Package 2 is really about gene bank operations, and Work Package 2 actually is led by Sipke Jamstra, so we are in the same family here. And also the colleagues from Boku, with Beate is here, also are involved in Work Package 1. Work Package 3 is the work package where colleagues are working on reproductive biotechnologies to enhance the quality of the reproductive samples that are stored in gene banks and to improve their usability, if it can be said in English, on the field. The work package 4 is the one about genomics, where we will use genomics to assess the potential of genetic diversity present in the collections. And the work package 5 is more a data work package where we want to set up a portal to facilitate access to information. And work package 6 is a work package about methods, quantitative genetics and population genetics methods, in order to propose new scenarios or to test strategies to make use of gene bank collections for the livestock sector. And the work package 7 is also a rather usual work package in any project, the one about dissemination, training, awareness, and including our ambition to contribute to an update of the guidelines for cryoconservation, the one published by FAO already some years ago. So we have a range of scientific disciplines in this project. So social sciences, because we need social sciences also to work with the legal bodies, with the stakeholders. We also have management sciences, economics, and this will be on the topic of the dialogue forum we have in Zagreb in August, what economics can tell us about gene bank operations. Also, we have colleagues in biology of reproduction, cryobiology, cell biology, mainly in the work package 3, and genomics, population genetics, biogeography, work package 4, informatics and bioinformatics in work package 5, and mathematics and quantitative genetics in work package 6, and communication, education science in work package 7. So here I have a very simple drawing to illustrate our question. So we have genetic diversity of on-farm population of a given species, total diversity in the A set. So if I can, it doesn't. Yes, you see this A, and in gene bank collection we have the B set, and you have the C overlap between A and B. And so the question is different for different species or different countries. What are the relative volumes of A, B, C? And A usually is used to update collections, also to store control samples in order to monitor trends, and also it is mainly stored as an insurance, as a backup. This is very often motivation. So A is providing diversity to B, and then when you still have very similar resources, C, the intersection can be used to restore a resource in case of a disease, of an emergency case. Then you still have the same diversity in the gene bank. But moreover, you have a diversity in the gene bank that may have disappeared from the on-farm population. And that's what we want also to look at. What is the best benefit we can draw from this old resource? Some people could think that it is so old that we don't need it anymore because we have genetic progress and this is old-fashioned and so we don't need it. But we can reintroduce lost variants and regenerate more viability in current population by using old collections, and sometimes also use them to combine between breeds and do like plant people do with pre-breeding when you test different genetic combinations. So to fill the gap about the knowledge about gene banks, so there was a survey set up by IMAGE about both germplasm, so reproductive gene banks, and DNA banks, which actually are poorly connected. You have a lot of laboratories with DNA samples related to the same populations which are in the gene bank, and this information is not connected. Regarding the collection, we also have our big five in Europe, cattle, sheep, goat, horse, and pig, which make the most of the collections, and altogether 546 breeds were found in the survey of the gene banks. More than 60 gene banks answered. But poultry and fish are increasing, and there is little redundancy across gene banks because they are nationals. But the major weakness is that less than 20% of the collections have molecular data, except for cattle. But for all the other species, the molecular information is really limited, and sometimes in chicken we cannot merge the data because they have been produced with different tools. So IMAGE will fill this gap by producing new data, additional sequencing and genotyping. You can see about chickens, pigs, cattle, and sheep. And we plan to develop a multi-species genotyping tool so that the aim would be that each gene bank could use, in any country, could use the same tool to assess the diversity of the collections and make it comparable with the diversity in other gene banks, just because they could use the same tool, so that it's much convenient to compare the results. So this means that we want to extract new knowledge from genomic data, both by genomics and bioinformatics. So there is, we are working on a conceptual model to connect all available data on a resource, the genetic data, the geographic data, the phenotypic data, and the starting point is a database of the EBI in Europe, the biosamples. If you have a unique identifier of a sample, then you can connect all the other information you have about this sample. And we are developing also a new pipeline to analyze sequence variation and a new method to identify a functionally relevant variation on genomic data. Population genetics, so my colleagues also have developed a method to detect deleterious variants, which is important to know when you are storing resources. And for biogeography, one Swiss partner has developed a method of environmental clustering where you can characterize the climatic or socioeconomic characteristics of different regions, and then you sample your animals according to these environmental clusters to have a representative gene bank. So other knowledge gaps are related to reproductive biotechnologies. There is still a great variability in reproductive efficiency of germplasm material. And so the answer is to improve sperm cryopreservation methods, particularly in poultry, where at the moment it's very difficult to predict fertility of poultry frozen semen. And there is a method developed by proteomics, and also in cattle there is a work done on microRNA related to differential fertility. And in some species we lack an efficient tool for complete recovery of germplasm, so we also have groups working to improve the embryo freezing in pigs and to develop alternate methods in poultry, because we cannot freeze the embryo in poultry. So colleagues are working on primordial germ cells or gonad grafting. So for gene bank operations, we identified a few very strong constraints. The first one being animal health regulations for exchange of germplasm, because these regulations are developed for large populations and for commercial trade, but the local breeds, very often they cannot afford heavy monitoring programs, and this may limit collection enrichment, because you are not always in the standard conditions to collect animals. And at the other end of the story, you can also realize that you have old samples that were collected at a period where regulations were different, and maybe you are lacking information on a disease status, because 20 years ago this disease status was not recorded or was not mandatory. And so we need to solve this issue in order to use all the materials. So all this may limit the use of collections. So this was a hot topic we discussed last year in our dialogue forum. And so we have initiated an action. The different partners are working with their corresponding experts in animal health groups at the level of the animal health law, which is under revision, or a new version is being prepared in the EU. So we are advocating the recognition of specific needs for animal gene banks in this framework. Other constraints about gene bank operations are the quality management. To build trust with users, quality management is a good answer because then you have traceability and standard procedures and you show your operations are sustainable and that it is worthwhile trusting you. But of course, this is an additional work and constraint, so we have proposed a diagnostic tool so that each gene bank can make a self-evaluation of its quality management. And FAO is currently using this tool for a worldwide survey ongoing. And training approach will be developed also for quality management. And the Nagoya Protocol, this I will not tell you too much, at least Ella Martignoud is in our project, so she is our specialist. But of course, we are working on benchmarking procedures and training because it can be very important for the use of gene banks. And the more innovative maybe aspect that colleagues from economic science have taken the gene banks as a new object and working on an economic model for gene bank operations. And what is interesting that these people are not at all involved in the usual operation of a gene bank. So they look at gene banks with a fresh or a new eye, the eye of an economist. So it is a bit challenging sometimes for gene bank managers, and that this will be discussed in Zagreb in the next dialogue forum. So to enhance the use of collections, as I told you, other colleagues are developing new methods. For instance, how to take into account the variable rates of inbreeding across the genome when you are optimizing a program to manage genetic diversity globally. There is also a simulation software that has been developed to model and optimize conservation schemes using ex situ conservation for in situ management of population. And we also have a demonstration project, a method how to plan an integration of a rare variant in a commercial population, and this is tested in laying heads with a rare variant about eggshell color. So the conclusion that it is a multidisciplinary project and effort, so we combine all these disciplines to produce new data, new tools, and in order to foster new ideas to manage and benefit from gene banks. So this is a long-term effort because this is only a 4-year project. And so, of course, this is done in the framework of the UGINA network of European animal gene banks, which provides a framework for continuation and taking advantage of the results of this project. And just to show where are our 28 partners, so they are in 13 European countries, but also because now in this call we were allowed to have other countries outside Europe who are getting also some funding. So we have Egypt, Morocco, and Colombia and Argentina, and we focused on countries with a rather hot climate because we thought that adaptation to climate change is a high priority for livestock, and that it was interesting to integrate these countries into our different work packages and different studies. So I will leave this here so that you have the picture of our coverage. Thank you for your attention.
Michèle, I think if we could, we'll just postpone our questions till after Sipke's presentation, and then we'll open it up for questions.